Hello everyone, welcome to Venper Academy. I hope everybody has done your final mock test, the grand mock test today. So I hope everybody has done it very nicely. So the questions are given over here and we are going to see the questions and answers. Okay, let's proceed on. So what's the first question? The small specialized roots in cycles are associated with which of these following? So the option A is given nitrogen fixing mycobacterium. Option B is water absorption by glomus species. And option C is nitrogen fixing filamentous cyanobacteria. And the fourth option is going to be nitrogen fixing filamentous fungi. Okay, first we have to understand what is cycles. So you know cycles are going to be what? Gymnosperms. So they are gymnosperms, which means they are mate-seeded plants. They do not have any kind of coverings over there. So they are gymnosperms. So then in case of cycles, if we have to talk about, they also have roots. These kind of plants also has roots, and these roots are in association with some kind of bacteria. And what is that bacteria? You can see some bacteria which are actually present over. And these bacteria are called as cyanobacteria. And what this cyanobacteria is going to do, because the plants cannot take the atmospheric nitrogen as such. So they have to be somebody else, or we can say the microorganism has to help them in fixing the atmospheric nitrogen in the soil as nitrite and nitrate. So that work has to be assisted by a bacteria called cyanobacteria. So we can say a small specialized root in cycles are associated with cyanobacteria, that too a filamentous cyanobacteria. So you can remember nostoc is going to be one such example where you're going to see the filamentous cyanobacteria. So the answer to this question is very simple. It's going to be option C. Okay, let's mark it. Option C. Okay. The next question will match the column 1 with column 2 is given here. So the first one is metagenesis, metastasis, and then metamerism, and the last one is metaphase. So everything is meta. Please don't get confused. It's very simple. You can literally answer this question. Genesis means formations. So we might have studied gluconeogenesis, all these things, right? So genesis means formations. So you can see formations of what? Yes, let's see that. So metagenesis, where you will see metagenesis, you have to remember. Okay. And then stasis, we will study about metagenesis. Okay. And metastasis. Metastasis is a condition which actually takes place in the cancerous cell. Okay. Suppose let me take an example. Okay. Suppose let's take a person is suffering from breast tumor. So if a person has a tumor in the breast, okay, if it is localized only in the breast area, we used to say it as benign tumor. They would not spread from one location to another location. But if the tumor, suppose let's take this is a tumor, which is going to be benign tumor. And suppose if this benign tumor is going to spread from the breast area and it's going to reach every part of the body through blood, then that is going to be malignant tumors. So malignant tumors are the one which are the spreaded form of a tumors. So the process is going to be metastasis. So which moves from the location where the tumor is present and it can go to different regions. So that's going to be metastasis. So let's see the property of malignant tumor. Can I say yes? So you have to see second is going to be 4. You can see only one option is going to be second is equal to 4. Okay, let's see the next one. Metamerisms. Okay, this we studied in animal kingdom, right? Yes, metamerism means segmentations. Metamerism is segmentations, right? So where do we see segmentations in an animal kingdom? We used to see segmentations in animal kingdom and very specifically in 3 phylums, if you can remember, Annelida, Arthropoda, and then Chordata. In Chordata, if you're going to talk about what will column is present, which is the internal segmentations. The rest of the things are external segmentation. So metamerism means segmentation. So what might be the answer? Segmentation marked distinctly. That's going to be the answer. So this will be 1. So this is also correct? Yes. Okay, let's see metaphase. So metaphase you will see in cell division, especially in mitosis. In mitosis, you'll be talking about prophase, metaphase, anaphase, and telophase. So metaphase, what exactly happens? The chromosome will be coming in the equatorial positions. So what we can say, the metaphase is going to be this one, right? In one we can say. So this is going to be the chromosome aligned at the equator. Okay, now metagenesis is going to be what? Alteration of generations, where you're going to see haploid situation getting converted to diploid, diploid changes into haploid. So haplodiploid. So metagenesis is the formation of a new generation by altering between them. So what might be the answer for this question? Very simple. It's going to be option D. Very simple, right? Okay, listen. So the next question, which stage of meiosis is recognized by dissolution? Okay, very simple. Suppose in examination, if they ask you dissolution, how can you remember this? Because we used to forget this. So suppose if it's going to be synaptonemal complexes. So what is the synaptonemal complexes? Let me write it. Suppose if you're going to talk about meiosis, we know meiosis is the cell. Suppose let's take a reproductive cell. A reproductive cell. So I'm writing reproductive cell which involved in reproduction. So suppose in case of male and in case of females. So males are going to have what? Sperms. They're going to produce sperms. So where is the sperm coming from? The male reproductive organ and the female reproductive organ. So they are going to produce what? Egg. Right? So this is a diploid set. So we can say this is a diploid set and this is a diploid set. Okay, and ultimately they are going to produce haploid set. So meiosis is the formation. We'll be studying about meiosis 1. We already studied meiosis 1 and meiosis 2. It's not mitosis, meiosis 2. Very important, you have to remember. Meiosis 1 is different, but in meiosis 2, you'll be seeing all the steps similar to that of a mitosis. Whatever you've seen in mitosis, you'll be seeing the same in case of meiosis 2. Okay, what is the synaptonemal complexes? Let's take this is one chromosomes. And let me take another color and let's consider that's another chromosome. Okay, let's take another chromosome. This is another chromosome. Suppose this is a paternal chromosome, this is a maternal chromosome. So here what exactly happens? Both are going to be crossed with each other. The genetic material is going to be crossed with each other. So that process is called crossing over at the synapses. The area where they exchange genetic material is called synapses, and the crossing over takes place. When the crossing over takes place, there will be a complex. The synaptonemal complex is nothing but it's a protein structure. That's all. It's a protein structure. So they will just come and form between them. Okay, this you already know. This is going to be sister chromatids because they are present in the same homologous chromosome. This is also going to be sister chromatid, but it's actually taking place between one chromatids of one chromosome, another chromatid of another chromosome. So the crossing over takes place between non-sister chromatid and there's a formation of synaptonemal complexes. The main thing is you have to hold the two chromosomes together. So you need a protein which holds them together. That's the role of synaptonemal complexes. Okay, yes. Now the question is, when will the synaptonemal complex is going to dissociate? Okay, when the genetic material has exchanged between them. So this genetic material will go here and this genetic material. So how the structure will be? Suppose they used to be getting. Let me write it here. Okay, let me just write it over here. So the genetic material. Suppose let's write here. So the genetic material will be exchanged here and the genetic material will be exchanged over here, right? So what exactly happened? Now they have to separate. So this phase, usually we already know everything takes place in prophase of the prophase 1. Very specifically, it is in the prophase 1. Prophase has different stages. Leptotene, yes, leptotene, zygotene, yes, zygotene, pachytene, yes, diplotene, and then diakinesis. Okay? Finally, it's going to be diakinesis. Remember, in zygotene, there's going to be fusion of two homologous chromosomes. Usually, if you're forgetting that, you can remember like zygote is a formation of male and female gametes. So fusion is going to take place where the crossing over takes place. In pachytene, what exactly happened? The synaptonemal complex is formed, or it's going to be dissociated. It starts to get dissociated. It is going to enter into dissociation. So in pachytene, there's going to be crossing over and there's going to be exchange of genetic material. Only in the diplotene, the dissociation will take place. Okay. So just remember, like in the diplotene stage, the synaptonemal complex is going to break. So you can remember like B and D. So in the diploteam, there's going to be dissociation where that synaptonemal complex is going to break between them, which is going to dissolve between two homologous chromosomes. So what might be the answer? The answer is option C. Okay, yes. So let's move on to the next question. So Sedaro-Okayo enzyme, which was helpful in polymerizing RNA with a defined sequence in a template independent manner. The important one. What's this template independent manner? We already know, suppose just a minute. So we already know there has to be a template and that there is no template also. Suppose if we are talking about DNA replication or DNA transcription, we know that there has to be a DNA. So one is going to be a template strand. So 3' to 5' is going to be a template strand. Suppose if I'm talking about transcription. And this strand is going to be just going to give rise to an mRNA, which is 5' to 3', which is mRNA. So usually there's going to be 2 strands in the DNA. So this strand is going to be 5' to 3'. And this is DNA. And this one strand is going to act as a template strand. So what are we doing? We are having a template strand and we are going to synthesize a new mRNA. So it is a template dependent one. But the question is, template independent one is asked here. So how can we remember template independent one without the template? There's going to be synthesis of this. Okay, now let's consider the mRNA is synthesized. mRNA, you should always remember it starts from 5' and it goes with 3' direction because hydroxyl group is present here. Right. So what exactly happened? 5' to 3' directions. So here we have already done something called a splicing and we make the DNA or RNA, sorry, it's RNA stabilized. How can we stabilize this? We add something called, we used to do a process called polyadenylation. We used to add a lot of adenine residues here so that this hydroxyl group, which is free, will not react with any other molecules. So that is called as polyadenylation. So are we using any kind of templates here? No, we don't use any kind of template. After the mRNA is synthesized, we literally add the enzymes to add this adenines and cations, all this thing. So it is a template independent manner. So the person is Severo-Okayo. So the enzyme is called as Severo-Okayo enzyme. What is polymerizing RNA? So we will add nucleotide. Nucleotide can be, here it would be ATP, GTP. Suppose if it's a DNA, we will say DATP, DGTP, DCTP, DTTP, all this thing. So what's the enzyme? So it is polymerization, which means you're going to add phosphates. So there has to be phosphorylase enzyme. Yes. Polynucleotide you're going to form. So it's going to be option B. Okay. Yes. So we'll see the next question. So in oogamy, very important, oogamy, fertilization involves what of these? We already know what's a fertilization, where the male gamete and the female gametes are going to be fused with each other. So we already know the male gametes are kind of smaller. Suppose let's take, they are going to be sperm. Yes, usually they are sperm, right? So if they are sperm, so sperms are motile, can I say? They are motile and they are small. Okay. And in case of females, we can say the egg is very large and they will not move. So the egg is large and they are non-motile. If the fusion takes place between the small motile sperms and a non-motile large egg, then it is going to be oogamy. Okay, yes. So what might be the answer? A large non-motile female gamete and a small motile male gamete. So answer is going to be option A. Right, yes. Okay, let's see the next one. The sequence that helps in identifying and eliminating non-transferments and selectively permitting the growth of transferments in the vector. This is the question from biotechnology. Okay, let's understand this. Okay, what I'm going to do, I'm going to take a plasmid. So where will I take the plasmid? We already know plasmids are coming from bacteria. So bacteria, suppose if I have to talk about the bacteria, suppose this is going to be the bacteria. The bacteria has nucleoids. They do not have a specific nucleus organized. Organized nucleus, they don't have it. So they have something called plasmid, which is extra chromosomal. So I'm going to isolate only the plasmid. So plasmid used to have some kind of properties we already studied. They have markers. So you have to remember this is a selectable marker. So what's the purpose of this selectable marker? So I'm just considering these are antibiotic selectable markers. Let's take, it's a tetracycline resistant gene is present here. Tetracyclic resistant, which is nothing but antibiotic resistant gene. So if you're going this plasmid and you're going to introduce into another bacteria, When you grow the bacteria in tetracyclic, because of the presence of tetracyclic resistant genes, they would not die in the medium. Usually, what's an antibiotic? We already know. And if you have a fever, the doctor used to suggest antibiotic. So antibiotics are actually prepared from bacteria, which is used to kill other bacteria. Yes. So antibiotics usually kills bacteria, right? Yes. So they kill bacteria. But since this plasmid is going to have resistant, even if you provide tetracycline, they can live. So that's an indication that your gene is gone inside or not. Let's take. So I'm going to introduce a gene. Suppose let's consider this is going to be a gene 1. Okay. So or you can remember like there's a plant which gives a high yield of crops. So that gene I've introduced over there. So this is called as a recombinant DNA. That's why we used to say genetic engineering or recombinant DNA technology. And now this recombinant DNA is introduced into a E. coli. There are many E. coli. And then we are going to introduce this plasmid into this E. coli. Many plasmids will be introduced inside. And let me just give only red color here for selectable marker and blue color for this new gene. Okay. So now what exactly happened? This plasmid will get inside. Not only one, so many plasmids will be given, all will get inside. Now I have to find out whether this gene 1 has got into the bacteria or not. So how can I find out? So there are two cases. So suppose if the plasmid containing gene, plasmid and the gene have got inside this E. coli, then they are called as transformants. They are called transformants. If the plasmid has gone or nothing had gone, the plasmid has also not gone, no genes had gone, then they are called as non-transformants. Okay, now how can we differentiate a non-transformant transformants? Suppose if a gene is going to express, ultimately this gene is also going to express. So what I will do is I will take this transformed cell and a non-transformed cell. I'm going to grow them in a medium containing tetracycline. So I'm considering transformed cell. So this is a transformed cell, this is a non-transformed cell. Suppose if I talk about the transformed cell, so this is a cell which has the plasmid and the gene. So let's use only blue color for this gene. So the gene is also present and then the tetracyclic resistant gene is also present. So this is a transformed. Now I'm growing them in a tetracycline antibiotic medium. So now due to the presence of tetracycline resistant genes in this, they can grow because they are showing resistant against the antibiotic. Antibiotics usually kills the bacteria. So they should have killed the E. coli. But since they have the tetracycline resistant genes, they will show resistance against the antibiotic. So this bacteria lives. So suppose if a bacteria lives, we'll come to a conclusion that they are transformed cells which has the genes introduced in them. Suppose if this is going to be another scenario where tetracycline antibiotic is given and the E. coli are grown, but here no plasmid, so no selectable marker, or we can say no tetracycline, no resistant genes, so the bacteria will be killed. Okay, so they die, they are non-transformed. So what is the selectable marker is going to be the one which identifies between the transformants and the non-transformants. Very easy. Okay. The majority of baculoviruses used as biological control agents. So what is that baculovirus are going to? It's a very important one. They are nucleopolyhedroviruses. Yes, you have to remember. They are nucleopolyhedroviruses. They are usually used as pesticides. In biotechnology application, you might have studied. They're used as pesticides. Suppose in a crop, there are a lot of moths, there are a lot of butterflies. If there are a lot of butterflies, they used to damage. There are a lot of moths they used to damage. So what they will do is they used to introduce this baculovirus into the farmlands. And ultimately what they will do, the genetic material which are present in it, right? They will just kill the moth and the butterflies. So it's a nucleopolyhedroviruses. Okay, the next question is a simple question, which is a question from photosynthesis. Okay, we already know about the C4 plants. Okay, in C4 plants, what exactly happened? We are going to talk about two things. One is going to be mesophyll cells and one is going to be bundle sheath cells. Okay, bundle sheath cells. So the question is, where is the A granule? A granule means A means absence. Absence of grana. Absence of grana. So what is a grana? Grana is nothing but many thylakoids. If there are many thylakoids are present and many thylakoids make up a grana. Suppose let's consider this is a chloroplast. I'm going to draw only the agranule and we will differentiate between the granule chloroplast. Okay. Suppose let's write agranule one. So this is the outer membrane, and this is going to be the inner membrane. And this is going to be the thylakoid. Only one means thylakoid. If many thylakoids are present, we call it grana. And there's a stroma lamellae, which connects, right? Yes. And listen. So if they are present, this is a granule chloroplast. Granule, because the grana is present, right? Yes. Suppose if the grana is not present, then they are agranule. So granule chloroplasts are actually present in mesophyll cells. And bundle sheet, where you will see a bundle sheet cell, suppose this is going to be the leaf. And if I'm going to talk about this is the cuticle, and this is the upper epidermis, and this is the palisade parenchyma, and these are spongy parenchyma, and this is a vascular bundle. And around this vascular bundle, you're going to see this bundle sheet. So these are going to be bundle sheet. In bundle sheet, suppose if I'm going to talk about one bundle sheet, and if you're going to see one bundle sheet, they are going to have what? Chloroplast. You write it. So they're going to have chloroplast. Suppose let me see one. There's going to be chloroplast, and this chloroplast do not have grana. And because of that, they are called as agranule chloroplast. So where is the agranule chloroplast present? They are in the bundle sheet of C4 plants or C3 plants. Can you just tell me? Yes, it is going to be, we can say it's option C4, right? It's option B. Okay, next. Okay. The next question. Elements involved in the synthesis of DNA. Yes. So whenever a DNA replication has to take place or a DNA synthesis has to take place, it's very important that if a DNA synthesis has to take place, DNA dependent DNA polymerase will come. If there's going to be RNA, they are depending on the DNA, so they are DNA dependent, and they're going to synthesize RNA, so they are DNA dependent RNA polymerases. So if in order to synthesize the DNA, you need DNA polymerase enzyme. And to synthesize RNA, you need an RNA polymerase enzyme. Yes, so these are enzymes. Right? So enzyme is always inactive when they are not bound with any cofactors. I hope everybody remembers APO enzymes. APO enzymes are inactive enzymes. An APO enzyme becomes a hollow enzyme only when APO enzyme plus a non-protein part bind together. So that non-protein part here is going to be what? Magnesium. So suppose let's take RNA polymerase. RNA polymerase binds with magnesium, then only they become hollow enzymes. Okay, so for a synthesis of DNA of RNA, you need what? Magnesium, very important. Okay, yes. So which of the following is associated with the regulation of sexual behavior or expression of emotional reactions and then motivation? Very important. What is this corpus callosum? Where is this corpus callosum present? Corpus callosum is actually present in the brain. Okay, and you know, this is the one which actually separates the right hemisphere and the left hemisphere. Suppose this is going to be the brain, and then there's going to be corpus callosum which will be present, which separates the right and the left. So they separate right and the left together, and that is corpus callosum. The question is, which is the sexual behavior? And you become emotional. What's the one which controls in the brain is the question. Okay, and what's the one which motivates you? Yes, you might have studied something called limbic system, right? Yes. So limbic system, you already know amygdala, and then hippocampus, thalamus, yes, thalamus, all this region. Yes. So thalamus is the one which relays information. And then hippocampus is the one which makes you remember things, memory. And then amygdala is the one which gives you emotions. So all these things together, we used to say limbic system. So answer is going to be option B. Corpus luteum, where do you see corpus luteum? Corpus luteum is the very important one because they are temporary endocrine glands. They are the one which stimulates what? Ovulations. They are the one which stimulates ovulation. So this is not the correct one. Medulla is very, very important, which involve in a lot of functions, temperature. They are involved in maintaining several other activities in our body. So here it's not the case. So we will not mark this option as correct. So answer is going to be option B. Okay. Which one of the following is put into anaerobic sludge digester? We have studied this anaerobic. Anaerobic means where there is no oxygen for further sewage treatment. Okay. So what we will do first, we will treat that is going to be activated sludge. After this activated sludge, we will transfer them to an anaerobic sludge digester where there is going to be anaerobic bacteria. So this anaerobic bacteria is going to react and they're going to produce methane finally, which smells, right? Yes. Usually methane is going to be produced. So answer is going to be option B. We will always take the first treated one and we will introduce them into the anaerobic sludge digester for further treatment, and ultimately you're going to get a kind of purified water. We are going to treat the sewages, right? Yes. Okay. Bicarpellary ovary with obliquely faced septum. What is bicarpellary? Carpels, very important. Carpels are pistils. So bicarpellary means we can say ovary is given. So ovaries will be like this. So bicarpellary means two carpels are present, like two carpels are present, and there are obliquely placed septum between them. So that you will see in solanum. You will always see in solanum. So answer is going to be option D. Okay, let's proceed on to the next question. Okay, so match the following column 1 with column 2 is given here. Okay, podocytes, protonephridia, nephridia, renal calculi. Okay, renal means kidney, calculi means calcium. So where it will be forming, it is in the crystallized oxalates. Very important. They are crystallized oxalate. Nephridia. So let's see this one. So for all the options are 1. Okay, let's see podocytes. Podocytes you will see in excretory systems where you can see them in the glomerulus and the Bowman's capsule region, where it is going to be like cells, and these cells are the one which pours all the filtrates inside. So they are the filtration slits, can I say? Yes. So 1 is going to be 4 and 4 is going to be 1. So we might be confused with option A and C. Reject option B and C. Okay. Protonephridia. What's the answer going to be for protonephridia? We can say yes. The second is going to be third one. So this is amphioxus. And nephridia, you already know it is annelida. So first is going to be 4 podocyte, and these are excretory organs. Protonephridia you will see in amphioxus, and nephridia you will see in annelida, and renal calculi you will see in crystallized oxalates. Okay. The next question, the nucleus. The nucleus is separated from the surrounding cytoplasm by a nuclear membrane is given. Okay, you already know what's a nucleus. And we are talking about prokaryotes, means the nucleus are scattered, or we call it nucleoid, which are associated with nucleoid associated protein and the DNA which is negatively charged. So if a nucleus, this is a nucleus and this is going to be the cell. The nucleus is separated from the cytoplasm by what? Nuclear membrane. And this nuclear membrane is double layer or single layer. Nucleus are going to be double layers. So they are double layers with pores or without pores. Yes, they are with pores. So answer is going to be option C. So we can say it's option C. Vaginal opening is surrounded by and covered by respectively. Yes, suppose this is going to be the vagina and this outer area is going to be the labia majora. Labia majora. Okay, or majora. And then there's going to be hymen, which is actually present. This is the hymen. Okay. So vaginal opening is actually surrounded by labia majora and then covered by hymen. So what might be the answer? It's option B. Yes. Okay. Next question. We'll move on to the next question. Okay. Which of the following approaches do not give the defined action of contraceptives? So what is contraception? Contraception is a pill which is used to prevent pregnancies after sexual intercourses, right? Usually. So barrier methods. What is this barrier methods are going to do? So barrier methods will not let the sperm and the egg to contact with each other. If a sperm and eggs are contacting with each other, ultimately fertilization will take place. So what are we going to do? We are going to prevent the fertilization. So is it a correct statement? But the question is, which do not give the defined action? So this can be the reason. Okay, let's see. So this can be correct. So we cannot choose this option. Intrauterine IUDs. In IUDs, what they will use is they use coppers. Intrauterine. Intra means inside. Uterine means able to keep the device inside the uterus. So they are going to use copper T tubes. And when they keep that, what exactly happened? Copper are very, very important. So if this copper is present over there, they would not let the sperm to move. And if a sperm is not moving, ultimately the activity or the motility of the sperm is reduced. And what exactly they're going to? They're going to engulf the sperms which comes through the vaginal region. Yes. So ultimately it will stop the fertilizations. So this is also a correct statement, but we cannot choose this option. Hormonal contraceptives. Yes. People used to take hormonal contraceptives to prevent what? Ovulations, fertilization. So this is also correct. But what about vasectomy? What is vasectomy? Vasectomy is usually done for males to be making them sterile. Okay. So what they will do, they will cut the vasa differentia portions and they will tie them, right? Yes. So whenever it's required, we can again fuse it back again. So what they will do, they do not want the sperm to go in this region because they are going to block the sperm entering through the vagina. Okay, so what are they going to do? Prevent spermatozoa, but not, we cannot say it's all the sperms are not going to be entering into the female reproductive tract. Some of the sperms will also be produced, but they are reabsorbed. So we cannot say they prevent the entire spermatogenesis, which means formation of sperms. So this is not going to be a complete statement. It does not confine to the defined action of contraceptive. So the answer is option B. You have to choose do not give. Okay, yes. The next question, which of the following make up more than one half of the neural tissue? If you're going to talk about the nervous system, the nervous systems are actually made up of what? Nerves. And we can say many nerve cells make up nervous tissues. But we can say if a person is having a brain tumor, so we know there are going to be neurons, which are nerve cells, and then there's going to be neuroglia. Neurons do not have, they cannot divide actually if you're talking about, but neuroglia cells are abundant. So they are the one which divides causing what? Nerve, we can say brain tumors like this. So which is the abundant tissue? Neuroglia, which means glia means blue, since it is present in the nervous, which holds the neuron tissues bound together. So answer is going to be option A. Okay, the next question. The prime contaminants of eutrophication. Eutrophication is a condition where you can see lot of deposition of some chemicals, which ultimately causes the aquatic living organisms to die, like marine life. Everything will be dying because there's a lot of more than nitrates and phosphate, which ultimately decreases the oxygen content in it. So the prime contaminant is nitrates and phosphates. Okay, the next question. Which of the following is correct about the hormonal actions is given here? What is this going to be? Okay, listen, where is this? Suppose let's talk about pancreas. So in pancreas, there's going to be islets of Langerhans. Yes, let's write islets. So in the islets of Langerhans, there's going to be alpha cells and beta cells. Beta cells are the one which produces what? Insulin. It's not glucagon. So they produce insulin. Insulin is very important because without the insulin, the glucose cannot enter into the cell. So insulin actually travels through the blood. Okay, so the blood, suppose if you're eating something, so the starch is converted into glucose, and this glucose has to enter into every cell. So glucose has to enter into a cell to produce ATP. Very specifically in a mitochondria, glycolysis, Krebs cycle, all these things will take place. So if the insulin comes and binds to the insulin receptor, then only the glucose can enter into the cell. So this is actually from beta cells. Insulin only is produced, it's not glucagon. So this is a wrong statement, but what they asked, correct statement. Secretion of thymosin is stimulated with aging. No, it's not the case. Suppose where is this thymosin are going to do? What's the role of thymosin? Thymosins are thymus. So something is related with thymus. So thymosins are actually produced from thymus. And what are they going to do? When a thymosin is produced, then a T cells are actually produced. Yes, but it does not get stimulated with aging. It's not the correct statement. So we cannot choose this option. It's a wrong statement. In females, the follicle stimulating hormones binds with a specific receptors on the ovarian cell membrane. Yes. So follicle stimulating hormone is the one which stimulates the follicle to get matured. Suppose if I'm talking, this is going to be the ovary and this is going to be the ovary membranes. And exactly in this region, there's going to be FSH receptor. And in this FSH receptor, this follicle stimulating hormone comes and binds. As soon as it binds, it will stimulate the follicles which are already formed in the ovary to form maturations, to undergo maturation. And then the follicles break open, right? Graphene follicles break open. So this is a correct statement. Okay, let's keep it as it is. Follicle stimulates the secretion of estrogen and progesterone. No, Follicle stimulating hormones will make the follicles to get mature and break open. It's not estrogen and progesterone. So again, it's going to be a wrong statement. The correct statement is going to be option C. Okay, the next question. To increase the volume of thoracic cavity or chambers in anterior posterior direction from up and down. Okay. So when do you want the thoracic chamber to be increased? When you're going to inhale, you want the space to be increased. If you want the space to be increased, the muscle has to contract. What is that muscle? The diaphragm. It is inspiration. We are talking about inspiration. So the diaphragm is going to contract. And even the internal intercostal muscles also contract. But the question is not given like internal intercostal muscle. They are given only external. So you cannot choose this option. So we will leave the first two options. Diaphragm muscles contracts. Suppose if a diaphragm muscles contracts, when they contracts like this, they open the thoracic cavity like this so that the air comes inside. So the answer is going to be option C. Okay. Identify the simplest amino acid. In our book, they have given very 4 amino acids, almost 3 to 4 amino acids are given. So we know what is amino acids. First, you have to remember amine is given, acid is given. So there's going to be one carbon and there's going to be one amine group, and then there's going to be carboxylic group. And then H is present because we have to balance it. And in this R group, if H is present, then we call it glycine. Okay, so answer is option A. Which of the following hormone act as a exocrine pancreas and stimulate secretion of water and bicarbonate ions? Okay, first you have to remember pancreas are going to be both. They have both the exocrine part also. They have endocrine part also. In endocrine, this endocrine is going to produce insulin, glucagon, but insulin is coming from beta cells. Glucagon is coming from alpha cells. Okay. Exocrine is going to release many enzymes, pancreatic enzymes. They also secrete secretins. And this secretin is very important for releasing water and bicarbonate. So answer is option A. Okay, yes. So the next question, which of the following process during decomposition is correctly described, which is very important. They ask the correct statement. So let's see. So fragmentation is carried out by organisms such as earthworm. Is it a correct statement? Yes. Usually the organic matters are broken down, right? Yes. So this is a correct statement. But let's see the next statement. Humification. So what is humification? You will see humus, the dark colored soil substances. It is mainly because aerobic bacteria will come and they will degrade all the organic matter into small inorganic matter, which is going to be a catabolism. And this will always take at a very slow rate. It's not very quick. It takes place at a very slow rate. But they have given fast rate, so you cannot choose this option. Catabolism is breaking down of large molecules into small molecules. Small molecules, like this. Suppose if you're talking about glycogen, which is stored in the liver and muscles, which is the stored form of glucose. So that has to be broken down into glucoses, which is going to be a monosaccharide. So answer is, this is also wrong. Leaching. Leaching is water soluble inorganic nutrient rise to the top of the soil they are given. What is leaching? When a nutrient or water soluble inorganic nutrient, because of rain, what they will do, they will go deeper in the soil. That is going to be leaching, which is again a wrong statement, right? Yes. So the correct statement is option A. Okay. Which of the following has both independent sporophyte and gametophytic stage? Sporophytic stage and gametophytic stage we studied in plant kingdom, right? Yes. So both are not dependent on each other. That you will see only in ferns, which is a pteridophyte. So answer is going to be option C. Okay. Let's move on to the next question. Which is correctly assigned? Yes, which one of these things is correctly assigned? Paramecium and Plasmodium belong to the same kingdom as that of a penicillium. They have given. No, penicillium belongs to fungus, but a paramecium and a Plasmodium belongs to protista. So this is a wrong statement, but we have to choose the correct statement. So we are not going to choose the statement. Lichen is a composite organism formed by symbiotic association with algae and fungi, not protozoa. So this is also wrong. So you cannot choose this option. Yes. Yeast, like Saccharomyces cerevisiae, you have to remember this. Okay. Saccharomyces cerevisiae is the one which is helpful to make bread and beer, and it's a fungus. Yes. And nostocs and anaconda are protists. They are given. No, they are not protists. Nostocs are going to be what kind of bacteria? They are bacteria. Anaconda species is a bacteria. So this is a wrong statement. So what is the correct statement? We can say it is going to be option C. Okay, next one. Choose the correct option is given here. Okay, it's very important. Staph fungi is called as ascomycetes. Ascomycetes. So neurospora is a ascomycetes. Correct. Imperfect fungi are called as deuteromycetes. Okay, which do not have the sexual reproduction taking place. Yes. And aspergillus is a ascomycetes. So it is not an imperfect fungi. It's not a deuteromycetes. So it's a wrong statement. Colectrichum. Colectrichum is also not going to be a bracket fungi or a club fungi. It is going to be again ascomycetes, so it is wrong. Phycomycetes they have given, and they have given agaricus. Agaricus is not phycomycetes, it is basidiomycetes. So this is also a wrong statement. So which is the correct statement? Option A. Okay, let's see the next question. Just a minute. Yeah. So choose the basidiospores are produced. Spores. Okay, spores are produced, which means they are haploid in nature. Basidiospores are produced, which means it is taking place in basidiomycetes in fungus. They are produced exogenously. Where is the spores produced? They are produced outside. Okay, so you have to see exogenous. First two options are given. And ascospores will produce inside. So it's going to be endogenous. So we can say option A is the correct statement. Okay. The next question, which of the following is going to be true about monocotyle? Okay, they're not talking about dicotyle. Okay, if it's a monocotyle, what type of venations you will see? Parallel venations, very important. You will not see reticulate venation. You will see reticulate venation only in the dicot, not in the monocots. Okay, so this is going to be a wrong statement. We cannot choose it, but we have to choose the true statement. Absence of bulliform is given here. No, absence of bulliform is present. This is also wrong statement. Bulliform will be present. Okay, bulliform cells are nothing but they are the cells which stores water. They'll be present on the epidermal layers. Okay, so they used to store water and they used to prevent transpiration, water loss. Okay, mesophyll cell is differentiated into palisade and spongy parenchyma. In monocotyle, do they have palisade and spongy parenchyma? Is it a correct statement? Yes, it is a correct statement. So what we can choose? Yes, we can choose the option C. Yes. The next question, stroma restricted to lower epidermis. Is it a true statement? Stroma is restricted to the lower epidermis only. In monocot, it is present in both the cases. Okay, the epidermis, suppose if they ask dicots, then you can say that in the lower epidermis, stomata is present. But in monocot leaf, you have stomata above also, you have stomata down also. So this is again a wrong statement. So correct answer is option C. Bulliform cells are present. Now we know what's a bulliform cell. They are mainly responsible for water surface, which are present in the epidermis, which prevents the water loss. Where it is present? Yes. So first you have to remember adaxial and abaxial. Adaxial, very important. Adaxial means what? Adaxial is in the upper epidermis. Okay. Abaxial is going to be lower epidermis. Okay, yes. So ab means bottom. Please remember. So bottom is lower, right? Yes. So abaxial. So now the question is, where is this bulliform present? Is it in the adaxial or abaxial? It is in the adaxial. Adaxial of monocot plant. You will see only in the monocot plant, not in the dicot plant. In the upper epidermis, you will see bulliform cell. So answer is going to be option B. Okay, yes. The next question, when huge amount of sewage is dumped into the river. Okay, suppose there's a beautiful river and there's going to be sewage introduced, all the ditch waters are introduced. So what exactly happened if the water is polluted? Ultimately, what happened to the oxygen level? The oxygen level gets less because other material becomes more. So we can say if the oxygen is becoming less, why will the oxygen become less? Because whatever the oxygen that is available, it will be utilized by the aerobic microorganism. Because aerobic microorganisms, let's consider aerobic bacteria. They utilize whatever the oxygen that is present and they will convert the organic matter that is present in the sewage into inorganic substances. Okay, so all the oxygen will be utilized by aerobic bacteria. So there's a deficiency of oxygen. So there's a demand of oxygen. So we say biological oxygen demand increases when there is pollution. So when a pollution is taking place, then the BOD increases. You have to remember. When a BOD decreases when there is no pollution, there's no demand for oxygen. So no pollution, BOD less. High pollution, BOD more. They are directly proportional. You have to remember this. So what's the answer for this question? It's going to be option A. Okay. The maize hybrids have developed for high amount of which of the amino acids? The maize, it's a hybrid. So it has been developed for high amount of an amino acid, which is going to be lysine. So answer is going to be option A. Let me write it. Yeah, it's option A. B. Lysine. Okay. Okay. Which of the following hormone plays an important role in RBC? RBC is the blood formation. It is called as poiesis. Poiesis means formation. Okay. So poiesis means formation. RBC we used to call erythropoiesis because erythrocytes. So they are erythropoiesis. So what are the enzymes that is needed? First, erythropoietin protein is needed. So you need erythropoietin protein. Yes. Where are you seeing this one? This one, this one, this one they are given. Thyroid hormones are important. Yes. So people who have thyroid, they used to have this anemia and all. So how it is related? Thyroid is related with RBC formations. So thyroid hormone is important, erythropoietin and cortisols. Cortisols are very important. Steroid hormones are very, very important. So answer is going to be option this one. Okay. So let's write it. So this is option A. Okay. Next question. So activation of pro-carboxypeptidase into carboxypeptidases. First, we have to understand what is this pro-carboxypeptidase? Where is it coming from? Okay. Yes. So pro-carb, you have to remember pancreas. In a pancreas, there's going to be enzymes which you have to remember like plant C. Just a mnemonic for you. Remember plant C. P refers to pro-carboxypeptidases. Okay. Pro-carboxypeptidases. And L refers to lipases. A refers to amylases. A is amylases. Amylases. And N refers to nucleases. And then T refers to trypsinogen. So examination, they would be asking you where is the trypsinogen produced. Don't get confused with pepsinogen and trypsinogen. Trypsinogen is actually coming from pancreas. Okay, from the pancreatic juices. C is going to be chymotrypsinogen. So now the question is, this pancreas, pancreatic juices will be poured into the duodenum. So suppose this is the duodenum, small intestine. So this juices comes over here. So the duodenum is going to be an intestine. They also secrete something called intestinal juices. And this intestinal juices, one such is going to be enterokinases. Enterokinases. And this enterokinase, what they will do is they will activate trypsinogen. They will activate trypsinogen. Trypsinogen is an inactive form. They will activate trypsinogen into trypsin. So now what this trypsin will do is, this trypsin activates all other pancreatic enzymes. So the question is, activation of pro-carboxypeptidases into carboxy. Pro means which is not activated. Into is brought about by trypsin. Okay, so answer is option D. Very simple question. Option D. Okay, the next question. The number of cockroaches, nymph of cockroaches. Nymph is going to be the young one of a cockroaches. So how many times it undergoes molting? Molting is nothing but the shedding of its wings or its exoskeleton structures. So it usually undergoes 13 molting. So they undergo 13 moles. So this is a very simple question. Okay. Suppose if we're talking about a plant growth hormones, it is responsible for horizontal. So suppose let's consider this is vertical and this is horizontal. So horizontal growth of the seed. Suppose this is going to be a seed. For horizontal growth of a seed, there is some hormone and the cell, or we can say the seed, actually swollen. It gets smaller now. This is also mainly because of one hormone, and they form an apical hoop at the tip. In dicot seed we are talking about. Which of these is important? Ethylene. Ethylene is very important for elongation and then for swelling and then for the formation of apical hoop in dicot seed. So answer is option A. Okay. Fruit ripener. Of course, ethylenes are used, but because ethylene has a lot of, we can say, harmful effects, we use something called as etiphones. So they can give you an alternative as etiphones. So where are you seeing? One is going to be 4. Okay, stress hormone is going to be abscisic acids in plants. When abscisic acid is produced, then the stomata will be closed when there is less of water. Stomata closing happens when abscisic acid is produced. Okay, so this is important. Herbicide is going to be 2,4-D. Bolting agent is gibberellic acid. So 1 is going to be 4, 2 is going to be 1, 2 is going to be, no, herbicide is going to be 3, and then third is going to be 2, and 4 is going to be 1. So we can choose the option as option C. Okay, the next question. The blockage of intestinal passage is a symptom of the infection. Okay, so people who have this infection, they used to have intestinal passage symptoms. There would be a blockage. It is mainly because of ascaris. Remember, please remember, it's mainly because of ascaris. Oxaloacetic acid, very important. Oxaloacetic acid is the first carbon compound which is formed, which is fixed actually. So what are they talking about? They're talking about the glucose production, which means they're talking about photosynthesis. In photosynthesis, they're talking about dark reaction. In dark reaction, what are they talking about? They're talking about oxaloacetic acid. First, you have to remember first stable compound. How many carbon does it make up? 4 carbon. It's a 4 carbon compound. So it is a C4 pathway. Now you have to find out what is the C4 plants. C4 plants, you have to remember it's going to be maize. So answer is A. Okay, select the mismatch pair related to the family. Okay, let's see. Mango is going to be Anacaceae, right? Wheat is Poaceae, correct. Housefly is Acidiae, correct. Cat is Candidae, no, they are Felidae. So they asked the wrong ones, so you have to choose option A. Okay, yes. So find out the true statement from the following. BSE is given. Okay, so first you have to understand what is this BSE. Let's write. It is caused by virions. They have given. No, they are not caused by virions. They are nothing but cow mad diseases. Okay, they are actually cow mad diseases, and it is actually caused by prions, not by virions. Okay, now you have to understand what is this virions and viroids. Very important. Viroids are going to be, they are similar to that of a virus, but they are going to be very smaller than that of a virus. And viroids, suppose if I have to talk about viroids, they have nucleus, or we can say very specifically, they have RNA also, they have protein also, the viral proteins also. In virions, you will be having only RNA, only RNA, only RNA. And in prions, you will be having only viral proteins, only viral proteins. So BSE is a mad cow disease or cow mad disease. It's actually caused by prions, not by this one. So they ask for the true statement. So you cannot choose this. Tobacco mosaic virus is caused by viroid. No, tobacco mosaic is caused by TMP viruses. So they are not viroids. Viroids are going to be what? Nucleus and proteins. That's not the case. Stunted growth is caused and curling of teeth is caused by viral. Yes, this is correct. Crown gall disease is caused by fungus. No, they are caused by bacteria. So this is a wrong statement. So you can choose only option C. So answer is going to be option C. Okay, next question we will see. Diadelphous stamens. Diadelphous stamens can be seen in citrus, pea plant, asparagus, and hibiscus. Okay, so stamens, you already know what is a stamen made of. It's male reproductive organs. Many stamens mix together to make a androgen. So a stamen has a filament, and then they have the anther. So let me write it. So they have an anther, which is bilobe. If there are going to be diadelphous situation, many anthers are fused. So these stamens are actually fused. Then we call it adelphous situation. Diadelphous means if the stamens are in 2 bundles. So fused, this is going to be many of the stamens are actually fused and they are in 2 bundles. Then we call this diadelphous stamens. And that kind of condition you will see in pea plant. So answer is option D. Glycosidic bonds. Where do you see glycosidic bonds? You will see glycosidic bonds only in the carbohydrates. So they have given adenine, insulin, inulin, trypsins. So first remember adenine is what? It's a nitrogenous basis. So it's a wrong statement. Insulin is a protein. So insulin you cannot see. Inulin is a glycosidic bond. So this is the correction. Trypsin is also going to be enzyme, which means they are proteins. So glycosidic bonds will be present only in the carbohydrates. So inulin is going to be the correct answer. Crossing of unrelated pure breeding animals of different traits within the same breed. So what are we going to do? We are going to take 2 different traits, different characteristics which are present in unrelated animals, which are not at all related. They don't have a common ancestors. And if we do, that is called what? Outcrossing, going beyond between different animals, brooding animals. That is going to be the correct answer. Okay, the next question. Choose the correct sequence of event that occurs in human reproduction. First, the gametes form sperm, and then the sperms are inserted into the female reproductive part, and the sperm and the egg will be fused together, and then the zygote is formed. The fertilization takes place in the fallopian tube of the female, and then the implantation takes place in the uterus, and then the gestation period comes, and finally the baby comes out, which is going to be the ejection of the fetus called as parturition. So answer is option B. Okay, which type of selection is industrial melanism? You already know about industrial melanism. We talked about this black color bark which has white color moths, and white color bark which has the black color moths. This is like mainly based on direction. There are directional type of sterilizations or selections we used to say. Okay, the next one, flying phalange and then a flying squirrel are example of what type of evolution they are. You have to remember this. You have to remember what is convergent, what is going to be divergent. Okay, so if we have to talk about convergent is like they don't have no common ancestor, but they have different same features. So you can see this is phalange is also flying, squirrel is also flying. So it's flying squirrels, and they have the same feature, but they do not have common ancestors. So this is going to be convergent evolution. Divergent evolution is a case which is just going to be the opposite. So they have common ancestor is present, but different features. That is divergent. Okay, adaptive, which is nothing but they adapt to the condition which is available, and they used to evolve. Okay, so answer is going to be option B. Colostrum, you have to remember it is IgA. So this IgA comes from the mother to the baby, through the milk. So if the baby has developed an antibody, if we are developing an antibody, we used to say that is active immunity. I am producing. Suppose if I get immunoglobulins, which is antibodies from outside to inside, that is going to be passive immunity. So the baby is getting immunoglobulins through the milk from the mother to the baby. So it's going to be passive immunity. Okay, next. 48. Bears, snails, zooplanktons, and seeds are given. Usually seeds, when they are inactive, we used to call it dormancy. And bears used to experience high, we can say it used to experience high cold conditions. Right? Yes, it used to exhibit high cold condition. At the time, what they want to do, they want to go in for hibernations, which is called as what? Very important. So they used to go to an area caves, and they used to protect themselves, and they used to have winter sleep. It's going to be winter sleep to protect themselves from very extreme conditions. And then snails are going to be aestivarians. So that is going to be what? Summer sleep. So when they feel very, very warm, okay, they used to go to water bodies, and they used to rely over there, and they would be in summer sleep. Okay, and then zooplanktons. What about zooplankton? Diapause situation. Diapause situation is an inactive dormant state that you will see in zooplanktons. Okay, yes, and seed is going to be dormancy. So what might be the answer? The bears are going to be hibernation. 1 is going to be 2, and 2 is going to be 4. 1 is going to be 2, and 2 is going to be 4. 3 is going to be 1, and then 4 is going to be 3. So the answer is option B. In Down syndrome, we already know about Down syndrome, which is a chromosomal genetic disorder, which we have seen in Principles of Inheritance. So here, the 23rd chromosome is going to have extra. So it's a trisomy conditions we used to say. This is 23. Usually they used to have 1 from the father, 1 from the mother. But here in this scenario, what exactly happens? Another extra 23 pair of chromosomes will come. So 3 chromosomes are present. So it is called as trisomy situation. So this is going to be option C. Okay, let's proceed on with the next question. Okay. So the sequence of nucleotide is given. By seeing this, you can easily understand. So UCG, CGA, and UGC is given. Usually, instead of thymine, if you see uracil, it's talking about RNA. So they said where you will find this. You will find only in RNA. Okay, suppose if there is an increase in body temperature, if the body temperature increases. So whenever a body temperature increases, the oxygen and the hemoglobin will dissociate. So what they will do, the oxygen composition will be more and the hemoglobin composition will be more, and the oxygen and the hemoglobin bound together will be very less. So if we have to draw a curve, oxygen dissociation curve, they will always shift to the right direction because of dissociation of oxygen and hemoglobin. So shift to the right. Okay. Suppose the state of heart when it is not pumping blood enough to meet the needs of the body. What is heart attack? Okay, that is called as myocardial infractions we used to say. Suppose that any part of the heart is not going to get sufficient amount of blood which carries oxygen. Any part of the heart, if they're not getting it, people used to get this heart attack. Oxygen has to be reaching everywhere, then only aerobic respiration will take place. This is heart attack condition. What is cardiac arrest? When the heart is not able to pump blood to supply oxygen, then it is going to be cardiac arrest. Angina is nothing but heart pain. They will be having severe chest pain. That is going to be angina. Heart failure is the situation where the heart is not able to pump blood effectively in order to reach the entire part of the body. Here, cardiac arrest, let me explain it again. Let me write it again. Okay. So heart attack is myocardial infarction. What exactly happened? Part of heart doesn't receive blood. Some regions of the heart doesn't receive blood, or we can say oxygen is not been received. Cardiac arrest, what exactly happened? The heart stops functioning, just stops working. So that is why heart arrest stops. And then angina is a chest pain. Okay, chest pain. And then heart failure is the heart fails to pump. Failure in the heart to pump. So heart pump decreases. So what is the question here? So it's going to be heart failure. The failure of the heart to pump blood. Okay, yes. So it's option B. The next question. So in cockroaches, the arthroidal membrane. Where do you see? Yes, usually if you're going to talk about arthropods has exoskeleton, and these exoskeletons are called as sclerites. And suppose if they are present in the dorsal region, they are called pterygm and sternum and then pleura. So how are they connected? Pterygm, sternum and then pleura are connected by orthodial membranes. So this is going to be the orthodial membrane. So it forms the what? Joins the sclerites. So it's option C. Proteolytic enzyme. Pepsin is a proteolytic enzyme. Yes. Trypsin is a proteolytic, but you should remember pepsin is in the stomach. Pepsinogen is getting converted into pepsin. Trypsin is actually coming from the pancreas. They are coming as trypsinogen, and they are converted by you can say enterokinases into trypsin. Then this trypsin activates thymotrypsin. Okay, these two are pancreatic enzymes, and this is going to be gastric enzymes. Okay, yes, so answer is option C. The epithelium is made up of single layer. Epithelium is the outermost layer, and if they are made up of only one layer, the flat one, they are squamous. Squamous are flat one, and they are single ones. Okay, so they are simple squamous epithelium, which are important diffusion in oxygen and carbon dioxide exchange. So that you will see in walls of blood vessels only, because they are helpful in diffusion. You will not see in glands. Okay, you will not see in nephrons, you will not see in bronchioles. The plant hormone that promotes female flowers, which means they have only female reproductive part in cucumber. What is that going to be? It is going to be ethylene. You have to remember, it's going to be ethylene. Ethylene is the one which favors female flowers in cucumber. Okay, in humans, suppose if we are talking about humans, at the end of the first meiotic division, the male gametes or the male germ cells, which means they are diploid cells, right? They are the one which is going to produce sperms. So they are diploid cells, and this diploid cells differentiates to form what? Secondary spermatocytes. Secondary spermatocytes. Okay, they said first meiotic division, meiosis. They are talking about the meiosis. They form secondary spermatocytes. About 71% of the total global carbon is coming from the forest. It's coming only from the forest. So answer is option A. Okay, let's see the next question. Which Which of the following represent the maximum number of species in the entire global biodiversity? Ultimately, it's going to be fungus. So fungus are actually predominantly present. So it's going to be option C. Initiation of lateral roots. Lateral roots are the one which comes in the side, right? So this is happening in the dipart plant. It is mainly because of which of these things? Pericycle. It's going to be option B. Because of the pericycle, they are actually arising of this lateral roots. Okay. The next question, and you'll see a potato, right? You'll be seeing an eye. So the eye of the potato, tubers are what? Axillary buds or root buds or flower buds or shoot buds. Suppose if you're talking about a potato, they have an eye, and in that there's going to be roots arising like this. They are axillary. They are coming from the axillary region. So they are axillary buds. So it's going to be option A. Conjoint vascular bundle. There are two types of ones. So one is going to be conjoint vascular bundle. Here, what exactly happened? Suppose in this bundle, vascular bundles are made up of xylem and phloem. Xylem is mainly responsible for conduction of water, and phloem is mainly responsible for conduction of food. So here, the phloem is located outside, and the xylem is located inside. So we can say the phloem located on the outside of the xylem, right? So xylem is present here, so they are located on the outside of the xylem. So it's going to be option B. Suppose, let's take the next question. Fibroblast. Yes, fibroblasts are very important for clotting. And then macrophages are very much important for fighting against infections. Mast cells are also important for fighting against infections. Where are they present? They are present in the areolar tissues. Very important. You have to remember they are present in the areolar tissues. If I have to talk about bacterial cell, so usually suppose if you're talking about there are certain bacteria which lives in rocks, archaebacteria, all these things. So they will get attached to the rock, but how do they get attached to it? What are the one which makes them get attached to it? It is mainly because of fimbriae. It's not villi, it's not fibri, it's fimbriae. It's option C. Okay, yes. The next one. Which of the following statement is correct with respect to proteins? Yes, remember carefully, in our body, we have D amino acids. D amino acids, which means they will always rotate in the right direction. If amino acids are made up of proteins, and ultimately the right-handed helix are present. So it's option A. Alpha helices are right-handed helices. Okay. The next they are talking about clitoris. What is this clitoris? It's a very important one. So clitoris is a tiny finger-like projection which you're going to see at the upper portion of labia minora or labia majora. That is very important. It is in the labia minora. Okay, which is it's going to be the majora and this is going to be the minora. Okay. So the small, tiny projection that you see, the clitoris region, right? That is option B. Karyogamy is a fusion of gametes. So when a karyogamy will take place, after that a meiosis will take place. So karyogamy is the fusion of gamete, and then if a meiosis takes place, ultimately they are going to produce haploid spores. And the spores are produced exogenously. The same question we have seen before, exogenous spore formation. Here it is in the neurospora. Yes, you have to remember it is in neurospora. Okay, yes. The next question, choose the wrongly matched pair. Okay, let's see this question also. Wrongly matched pair. Carrot grass is given here. Okay, and then wildlife is given here, and then Amazon rainforest is given here, and then Kasi and GMT hills is given. Let's see. The common name of a carrot is not Lantana candra. It's a wrong statement. And then wildlife safari is the exit to conservation. Yes, it is correct. And Amazon is the lung of the planet or the lung of the world, the lung of the entire biodiversity. Khasi and Jaintia hills are actually present in Meghalaya, the northeastern part. So this is the wrong statement. So we can choose this statement. ATPase, which is an enzyme, are needed for muscle contraction. Yes, when a muscle is going to contract, they're going to pull the actins and myosins are going to be bound with each other, and they're going to contract the muscle so that the sarcomeres gets reduced. So ATPase, where it is present. Suppose if I have to talk about myosins. So in the myosins only, this ATPase, suppose if we talk about myosins, they have a head and they have a tail like this. So this is going to be the actin binding site. I hope everybody remembers this. This is myosin, this is the actin binding site. This is actin binding site. Right. Binding site. And this area is going to be the ATP binding site. ATP binding site means where the ATPase can come and bind. So where it is present, it's actually present in meiosis. Abrine is a question, very important. It is a toxin. It's a toxin. Okay. The first stable compound of nitrogen fixation. Okay. How will a nitrogen fixation will take place? The first stable compound formed is ammonia. Okay. So ammonia will be the most stable form of compound in nitrogen fixation. Okay. They have given a lot of fishes and you have to find out which one of these are edible fishes except, which means they are not edible. Common carp is a freshwater fish, but they are not edible. You cannot eat that kind of fish. So they are non-edible fishes. Hilsa you can eat, sardines you can eat, pomfrets you can eat, but this is not going to be the correct. Okay, let's see the next question. Diversity is the type of beaks of finches adapted to different feeding. In Galapagos Island, what Darwin was telling, they were a different kind of finches and they adapt themselves to form what? Yes. How do they live in that situation? They have different feeding habits. One eats grain, one eats another form of grain, one eats meat, one eats maize. So he said like they are adapting themselves to that environment based on the conditions. So it is called as natural selection according to Darwin. Darwin's theory, you already know it's natural selections. The visible colored portion of the eye is involved in what type of regulation? We can see the colored portion of the eye, right? The visible colored portion. Where is it present? It is the diameter. It is helpful in regulating the pupil. The pupil diameter actually increase and then decrease, right? Yes. So it is going to be option B. The life cycle is a very important question from plant kingdom. You have to study all the alteration of generations. Ectocarpus and fucus are, ectocarpus is going to be haplodiploidy. You can see haploids are also formed equally and diploidic situation also formed equally. So they are haplodiploidic and fucus is diploidic. So it's option C. Okay. The next question is which of the following suffixes are used to identify or to indicate category of order? They are talking about order, not okay. So order you will always say polynomials, ALES. So it's option C. Okay. In Cran's anatomy, which means they're talking about C4 pathway like maze. Okay, so there are going to be two type of things are involved. One is going to be mesophyll cells. In C3, only mesophyll cells will be involved. In C4, mesophyll cells as well as the bundle sheath also will be involved. So are they going to be thick walls or thin walls? Yes, they are thick walls. So suppose this is the vascular bundle, is going to be bundle sheath present. So they are thick wall and they do not have any spaces. Do they have any spaces? So they do not have it. And inside, if you're going to see, they're going to have 8 granule chloroplasts, but the 8 granule chloroplasts are going to be more in number. So the answer is going to be option C. Ladybird is useful to get rid of aphids. So ladybird, if it's present, aphids will not be present. Okay, yes. Select the incorrect statement is given over here. So bilirubins and biliverdins are the bipigments. Yes, they are the bipigments. So this is a correct statement. Emulsification of fats. Yes, the fats are going to be water hating molecules. So the water loving molecules comes and binds around this water hating molecules. So we used to call it missles. Yes, so that is also a correct statement, but the question is incorrect. So we have to find out which one of these is incorrect. Renin is a proteolytic enzyme. In babies only you will see, but it is found in the pancreatic juice. They have said no, it is actually in the stomach. Okay, in the stomach only this renin will be present. So this is a wrong statement. So we can choose only the third option as a wrong statement. Mucus and bicarbonates protects the mucosa. Yes. So when HCl is going to be highly acidic conditions, this mucosal epithelium, we know there are 4 layers: serosa, muscularis, submucosa, and mucosa. This mucosa layer secretes mucus and bicarbonate, and they balance the pH. So this is correct. This is only wrong. During lactic acid fermentation, what exactly happens? So the pyruvic acid is getting, which is a 3-carbon compound, it is getting converted into lactic acid, which is again a 3-carbon compound. A 3-carbon getting converted into 3 carbon, so no of carbon dioxide is released. The enzyme is going to be lactic acid dehydrogenase. So it's option A. Dense irregular connective tissue, where are you going to see? In the skin. Skin is going to be a dense connective tissue, but it is irregular. So it is option C. Okay, the next question. Consider the cross AA, BB, and AA. What you have to do, let me write the gametes alone, you can do it. Okay. Capital A and capital B, and capital A and small b, small a and small b, small a and capital B. So what you have to do, you have to do a tabulation of this. The unit square you have to do capital A, let me write it. It's going to be capital A and small b. Capital A and then small b, and then small a and then small b, and then you have to write small a and capital B. And if you're going to do a Punnett square, you can easily find out what is the genotype. So you can write how many AA, BB are present, how many small a, BB are present, and how many of them are present. So answer is going to be option B. If you do this one, it's going to be your answer. Okay. Which one of the following explains the role of heat shock? Usually in biotechnology, what we will do, we want to introduce the DNA into the host cells. So at the time, we used to give a heat shock along with calcium. So what they will do, the uptake of DNA will take place, or binding of the DNA to the bacterial cells, or decrease the efficiency of transformation. Usually, what exactly happens, the DNA will be taken to the pores. So we are creating a pores by giving a heat shock. So it is option B. Semi-conservative replication, you already know by Michelson and Stable's experiment. He demonstrated with which of these Drosophila melanogaster, Acacia coli, Streptococcus pneumoniae. Yes, it is. So which of these Acacia coli? 20 minutes, right? So it's going to be option B. In field experiment, when all the Pisastus starfishes were removed from the enclosed area, the result was, if you're going to remove one starfish, ultimately the other invertebrates will be dying. So there's always going to be correlation between two species. So it's going to be extinction of the other species. Vallisneria is an aquatic plant. In aquatic plant, pollination has to take place, which means the sperm has to fuse with the female reproductive part. So Vallisneria, inside this plant is going to be the ovum present. So the male gamete comes out of this another plant and they used to fuse with this one. So male flowers are carried by water current and to the female flowers where it is present. Okay, yes. The next one. What is the direction of movement of sugar in the phloem? Yes, sugars are the one which are, we can say food is actually transported to phloem. Okay. So in which direction it goes? The food has to go from the bottom till the top, and from the top till the bottom also it can come. So it is bi-directional. So answer is option B. Okay, a creeping green branch and a filamentous structure that you find in mosses. Okay, which means they're talking about bryophytes. That is going to be protonema. From the protonema only the plant actually arises. Okay. 89 question. Periderm, perisperm, pericarp, and perimetry. First, we will see perimetry. Metry means uterus. It's related to uterus, some layers. So perimetry is lining of the uterus. Pericarp is the fruit wall, the outer wall. And perisperm is the nucellus, persistent nucleus. Periderm is phellem, phellogen, phelloderms, all these things together, we say periderm. So what might be the answer? The first is going to be 3, the second is going to be 2, third is going to be 4, and fourth is going to be 1. So answer is going to be option D. Okay, yes. The mango and the coconut develop from which of these? Yes, they are monocarpals, which means they have only one carpel and they come from superior oval, not from the inferior oval. Okay, yes, which means they are hypogynous flowers they have. Okay, yes, it's option B. Gross primary, total, gross means total. The rate at which how many organic molecules like glucose are produced by autotrophs, which means one which produces one which perform photosynthesis to produce glucose. How much is the glucose is formed is going to be primary. Primary means primary producers is going to be plants, which is autotroph. How much of the organic material like glucose they are able to form? So answer is option B. Okay. A patch of nodal tissue responsible for initiating contraction of the muscle. Where do you see? Yes, you know, suppose this is going to be the heart and this is going to be present over there. And this is right atrium and this is left atrium and this is right ventricle and this is going to be left ventricle. So here there's going to be SA node. So where is it present when the SA node is going to get impulses? So it is present in the upper right corner of the right atrium. Can I say they are the one which helps these ventricles to contract? So it is option B. Okay. The next question. The food having mechanical chemical digestion in the stomach. Okay, the food is coming from the mouth and it's going through the esophagus and then it is going to the stomach. When the food has reached the stomach, it is partially digested. That time we used to call it chyme. Okay, bolus means when it is present in the mouth along with some kind of saliva and mucus and salivary amylase, we used to call it bolus. Anterior portion of the sclera is cornea. You already know this is going to be the cornea, the front portion, right? So which of the following is incorrect with respect to honeybee? Honeybees, male honeybees cannot perform sperms because they are parthenocarpic. They will not perform this one. So male honeybees produces sperms by meiosis is a wrong statement. This is not going to be possible. It's a wrong statement. Next, which one of the following is a part of pectoral girdle? Yes, glenoid cavity is going to be the part of pectoral girdle. This question has been repeated many times. So it's option D. Hollow bones. Hollow bones means they're going to have spaces that you will see in birds. So answer is option D. The next question, identify the substance which form glycosidic bonds. So you already know what's a glycosidic bonds. So glycosidic bond is between a sugar bonds, between sugar. And peptide bond is between amino acids and amino acids. So R, you can know that R, and then C, and then NH2, and then COOH, and then H. So another amino acid will combine together, which is going to be NH2, and then C, and then R2, any other compound, and then it's going to be H, and then H. So this H2 and then one oxygen combines together, and they will be released. So what are the bond you'll get? CO, NH bond, and the rest of the things are going to be same. This bond is called peptide bond. So where you will see peptide bond in a protein. So albumin is a protein, and sugar is going to be maltose. So cellulose is a carbohydrate. Rubisco is the enzyme. They made it this side. Okay. Cellulose is carbohydrate. Protein and carbohydrate. So this is only the right statement. Which of the statements about the papillonoceous aestivation is given? Okay. Yes. So what exactly it is? So here, how is the arrangement they have talked about, right? Petal arrangements, sepal arrangements. So papillaceous, you will see 5, right? One is going to be the standard ones, standard petals or standard sepals, and then the lateral ones, and finally you're going to see the keel. So what are the answers? Standard petal is largest petal, and wing petal is the smallest they have given. Smallest anterior petals overlap the lateral. The lateral petals is overlapped by the standard one. So the option is going to be option C. Okay, answer is option C. Yes. So the lateral petal, right? This actually gets overlapped with this one like this. So they will be like this actually. Okay. High calorie, low protein diet to extensive edema and swelling of the body. This will happen in marasmus conditions or in kwashiorkor. This will happen in kwashiorkor conditions. Okay. Now, what's the difference between a marasmus and a kwashiorkor conditions? Suppose if you're talking about in case of a marasmus conditions, it will take place in the kids only. Kwashiorkor will also take place in the kids, but there's a difference in the age groups. Okay, suppose if you're talking in case of a marasmus conditions, marasmus people, small kids used to have less than 1 year. Kwashiorkor, they will have more than that. Okay, that's the main difference. Okay, here in case of marasmus also the protein content is low, but kwashiorkor high calorie will be there. High calorie of the thing will be there, but the protein content will be very less. Because of that, the baby used to have the bellies are larger, and they have edema, and they will be mentally retarded. So answer is going to be option D. Okay, so we are done with almost all the questions. So I hope everybody has done the grand mock test very well. So exams are very close by. I would suggest you to revise well. Don't be stressful. Be calm. You're going to do it very well. All the best to all of you from the Venper Academy. Yes, thank you all of you.